Namaskar. Today we'll study Lac Operon. So Lac Operon is Lac stands for lactose. Lac stands for lactose. So lactose operon. And what is actually operon? So for that, let us discuss bacteria needs nutrients like your glucose, and when there is lack of glucose, and if we give lactose, from lactose it will obtain glucose. So lactose has to first enter into the bacterial cell. So this is bacteria. Lactose has to enter into the bacterial cell, and the enzyme required is polymerase. And lactose is a beta galactoside, so the enzyme can be beta galactoside polymerase. Then lactose is degraded to glucose and galactose by the enzyme beta galactosidase. So these two enzymes are required for the production of glucose from lactose. So Lac Z, Lac Y and Lac A, these are the three genes which are functionally related. Transacetylase is another enzyme which decreases your thiagalactoside level. So the bacteria gets rid of the toxic thiagalactoside because of transacetylase. Here for lactose, we need these two enzymes, beta galactosidase and beta galactoside polymerase. And beta galactosidase, the gene is Lac Z, and beta galactoside polymerase, the gene is Lac Y. So since these three genes are functionally related, they together function, and they have a common operator, they have a common promoter, they have a common regulatory gene, and the regulatory gene is called Lac I. So what happens in Lac Operon? This RNA polymerase we need because that will convert this DNA to messenger RNA by the process of your transcription. So DNA is converted to messenger RNA by transcription, then messenger RNA is providing the information for the required protein to be synthesized. So this Lac Operon, so when this RNA polymerase binds to the promoter, that will simultaneously express these three genes, Lac Z, Lac Y, Lac A, and will produce the respective proteins, the enzymes, beta galactosidase, beta galactoside polymerase, and transacetylase. And there is operator site to which the repressor binds. Repressor binds to the operator site, blocks the operator so that there will be no movement of the promoter, no transcription. And repressor protein is produced from Lac I. So Lac I is a constitutive gene which produces this repressor protein. It is always operating, and this repressor protein binds to the operator and inhibits this gene transcription. So normally the Lac Operon is off. Lac Operon is off because Lac I always produces this repressor protein, and repressor protein binds to the operator and inhibits the gene transcription here. But when there is requirement, so lactose is, if lactose is provided, then lactose binds to the repressor. So usually what happens, this Lac Operon functions mildly, so that lactose enter into the bacterial cell and will inhibit this repressor protein. Otherwise, it cannot function. So usually it is normally it is functioning mildly. So lactose can permeate into the bacterial cell and will bind to the repressor protein. And when this repressor protein is blocked, it cannot bind to the operator. So when it cannot bind to the operator, then that means RNA polymerase can move, and there will be a transcription of all these three genes, and there will be production of all these enzymes, and then lactose can easily permeate into the bacterial cell and then it can be degraded into glucose. So when all the lactose is depleted, there will be no more lactose. This repressor protein becomes free and can again bind to the operator, and there will be inhibition of this RNA polymerase action, and RNA polymerase action when inhibited, there will be no gene transcription. So in this way, when there is lactose is present, then there is gene transcription, and when there is no lactose, there will be inhibition of gene transcription. So when presence of lactose induces this operon, so this operon is also called as an inducible operon, because the substrate is here lactose, and when lactose is present, there is gene transcription. So this operon is known as inducible operon. So another site is there, and here lactose is degraded. There is catabolism of lactose. So this operon is a catabolite operon, or the genes are catabolite genes, and there is a catabolite activator protein site here. So when this catabolite activator protein binds here, this will facilitate the RNA polymerase action. RNA polymerase action is facilitated, and this catabolite activator protein itself is inactive. In presence of cyclic AMP, it is activated. So here you can see this site is not there, and when cyclic AMP binds, there will be this site which will bind to this RNA polymerase and will facilitate the RNA polymerase binding with the promoter, and it will facilitate the action of RNA polymerase, so that there will be gene transcription. So again, this cyclic AMP causes, cyclic AMP facilitates the binding of CAP and causes the gene transcription. So we can have two types of regulation. When it is regulated by the repressor protein, we call it as the negative regulation, and when it is caused by the activator protein, we call it as the positive regulation. So this regulation of this Lac Operon can be done by this activator protein as well as by this repressor protein. Repressor protein in a negative regulation, and this activator protein positive regulation. So how it happens? So LAC-I gene, the respective messenger RNA, and produces this repressor protein. Repressor protein binds to the operator and inhibits this RNA polymerase action. RNA polymerase binds to the promoter site, and RNA polymerase has to move in this direction, so that it will cause the transcription of all these three genes, LAC-Z, LAC-Y, LAC-A, and a messenger RNA will be produced, and the enzymes or proteins which will be produced, all these three enzymes will be produced, which can cause the degradation of lactose. So normally this repressor protein is produced and inhibits the operator's region and inhibits gene transcription. So normally this is off. And when there is lactose binds to this repressor protein, and when lactose binds to this repressor protein, it cannot bind to the operator site. So it allows the gene RNA polymerase access to the promoter followed by all these genes. So there will be transcription and the genes are expressed. So this is negative regulation. When there is no lactose, gene expression is off, and when there is lactose, there is on, gene expression is on. So here normally this is off and this is on. Positive regulation, when this Lac Operon is regulated by the activator protein and the co-activator, we call it as the positive regulation. So this catabolite activator protein is inactive, when the cyclic AMP, it has this binding site for cyclic AMP. When cyclic AMP binds to it, it becomes active and can facilitate the binding of RNA polymerase. This is RNA polymerase. Facilitate the binding of RNA polymerase to the promoter site, and it will facilitate the gene transcription of LAC-Z, LAC-Y and LAC-A to produce all these enzymes. So, for this, what happens? Normally this cyclic AMP is produced from adenylate cyclase. So, adenylate cyclase is required for production of ATP to cyclic AMP, and adenylate cyclase. So we can say active adenylate cyclase in presence of your high glucose becomes inactive adenylate cyclase. So in presence of high glucose, or when lactose is degraded to glucose, this adenylate cyclase becomes inactive and there will be no cyclic AMP production. So this operates only in case of low glucose. When there is low glucose or no glucose, then adenylate cyclase is active and cyclic AMP is produced, and this will facilitate. So when there is no glucose, we need glucose. So no glucose will activate the production of cyclic AMP, and cyclic AMP will do this positive regulation, and cyclic AMP will activate this catabolite activator protein, which will bind to the CAP site and will facilitate the action of your RNA polymerase. And when there will be glucose, again when there will be production of glucose, so that will cause no glucose, gene transcription, and lactose will be breakdown. So beta galactosidase causes degradation of lactose to glucose, and when there will be glucose, the adenylate cyclase will be inactive, no cyclic AMP, and again it will stop. So this is called your positive regulation, because cyclic AMP causes your gene transcription, and this is called negative regulation because this repressor protein inhibits the gene transcription. So in this way, this Lac Operon operates. Thank you.